Hello, today we are diving right into meiosis. This is just going to be a pretty brief overview of what happens in each of the stages, what meiosis is, just the important bits so that when we get into a little bit deeper into genetics, you understand why like genetic disorders happen, genetic diseases happen, and how things can get passed on from parents to offspring. So to start, let's talk about the purpose, and the purpose is the production of gametes. That is like, by and away, the baseline purpose of meiosis. It is to create sex cells. There's a lot of steps in meiosis, but the end goal is to produce gametes. Meiosis takes place in two sets of cell divisions, meiosis 1 and meiosis 2, and the steps within each of those divisions are the same, so it can get a little confusing. The end result here is that you have 4 daughter cells rather than 2 daughter cells that you would get in mitosis. These 4 daughter cells are haploid or only have half as many chromosomes and are genetically unique. So another important piece of information. In humans, we have 2 types of cells. We have somatic cells, which are like our body cells. This would be like a skin cell, a muscle cell, a nose cell, like anything that you would find in your body that is not egg and sperm is considered a somatic cell. So, body cells. And within our body cells, we have 23 pairs of chromosomes. So in any general cell, you have 46 chromosomes, which is broken into 23 pairs. The pairs are called homologous chromosomes. We'll get into that on the next slide. The other types of cells that we have in our body are called gametes, and these are sex cells. These are like your egg and sperm, right? Biological females have eggs, biological males have sperm. These have half the number of chromosomes, and so there's only 23 total chromosomes, so they're not in pairs, they're single chromosomes. Now, homologous chromosomes. We mentioned this on the previous slide, but this is pretty much like your basic pair of chromosomes. When you have a homologous chromosome, or homologous pair, they are the same length, they carry the same genes in the same area, and they control the same inherited characteristics. For all of our pairs of chromosomes, you get one from your mom and one from your dad. And so for every pair that you have one from mom and one from dad. And that's what gives you your unique look. You may look like one of your parents, but you actually do have a unique genetic makeup. Unless you're an identical twin. This is just an image of homologous chromosomes. They look like this, okay, right? The purple maybe is from dad, the green is from mom. When they replicate, they look like this, like the typical X of a chromosome that you would see. Each individual half of a homologous chromosome is called a sister chromatid. That will come in handy later when we are going through this, but just to give you an idea of what homologous chromosomes look like, they can be singular or the X shape. So, going into a little bit more detail with the chromosomes in human cells. So, when we're talking homologous chromosomes, when we talk about the pairs, right, there's 2, there's one from each parent, and these cells that have the pairs of homologous chromosomes are called diploid cells. That's a very important distinction. Diploid cells are like your body cells. The human diploid number is 46, right? We have 46 chromosomes. Our diploid number is 46. That is not the same for other species, but for humans, which is what we're going to focus on, our diploid number is 46. Because gametes only contain a single set of chromosomes, it is considered a haploid cell. So diploid is 2 sets, haploid is 1 set. The human haploid number is 23, right? This makes sense. 23 is half of 46. A haploid cell has half the amount of chromosomes than a diploid cell. And the types of chromosomes, I think this is the last bit before we actually get into meiosis, but types of chromosomes within our pairs. The first 22 pairs are called autosomes. Autosomes are just like your typical chromosomes that lead to your general information. Pretty much all the information that you could want about your body that are not found on sex chromosomes. Those 22 pairs are called autosomes. The 23rd pair is called your sex chromosomes. Pretty straightforward, right? The 23rd pair is the X and Y in humans. Biologically male humans have one X and one Y. Biologically female humans have two X's. We will get into some genetic disorders where this is not true. There are disorders where humans can have 3X chromosomes or 1X or XXY. There's lots of different disorders that can be caused from your sex chromosomes, and we will get into them, but this is just sort of like a brief overview of the general terminology that you will need to move forward. Oh, and fertilization. So fertilization is the union of the gametes, right? So when a sperm and an egg come together, it's called fertilization. The fertilized egg is called a zygote and has one set of chromosomes from mom, one from dad. The zygote produces somatic cells, your body cells, by mitosis, and that zygote will develop into an adult, right? So like any fetus starts off as a zygote. All right, let's talk about meiosis. So as you can see here, there are two divisions, right? This is the beginning, and then it starts to divide right here. This is division 1, and then you get two cells, and they each go through another division, and so this is your second division all the way at the end. So for this, your first set of like meiosis 1, you can see it has a 1 at the end of everything, and then the second division has a 2 at the end of everything. So we're going to talk about what happens in each of these, but realistically, the same thing happens in like prophase 1 and prophase 2. It's just that you are dealing with less chromosomes at that point. All right, so let's get started. So meiosis has two divisions, like I just mentioned. Meiosis 1 is when homologous chromosomes separate, and in meiosis 2, sister chromatids separate. If you're confused about what those terms mean, please refer back to the beginning, in which I showed you a picture that talks about the difference between the two. So interphase happens prior to meiosis. This is sort of where cells spend most of their life, like 90% of their life. And during this time, cells are sort of just like hanging out, doing their own thing, like whatever, living their best life. And right before they get into meiosis, the chromosomes duplicate and the centriole pairs duplicate. The centrioles are these like little yellow like mac and cheese looking things. They're going to be responsible for shooting out spindle fibers, and those spindle fibers will come in handy to move the chromosomes around where you need them. In prophase 1, your homologous chromosomes pair up. They form a tetrad when they pair up. So when you have 4 sister chromatids together, that forms a tetrad. I'm sure you've heard of tetrad before. If nothing else, I know you've heard of tetris. That prefix, tetra, means 4. Tetris, if you've ever played Tetris, the reason it's called that is because each shape has 4 blocks in it. And if you didn't know that, now you know. So in prophase 1 anyways, chromosomes coil up, they form into actual chromosomes. The centrioles, your little mac and cheese guys, move to opposite ends of the cell, and each chromosome appears as 2 identical sister chromatids joined at the centromere, which is this little like piece holding it together. The nuclear envelope dissolves, it starts to break up, and microtubules attach the centromere to the centriole. That's like your spindle fibers there. Okay, so big takeaways here is that homologous chromosomes form and they pair up, and the centrioles shoot out spindle fibers in so that they can get ready to start connecting to the chromosomes and start moving them around. In metaphase 1, this is the next part. You see these spindle fibers have shot out. They've connected to the centromere here, and these tetrads have lined up in the middle of the cell. This is like called an equator. It's exactly what you think of when you think of the Earth's equator. It's just the middle of the cell. And these centrioles have used these spindle fibers to move the tetrads into the middle. In anaphase 1, the spindle fibers pull along the centromeres, which are again these little like black dots in the middle of the chromosomes, and they separate the homologous chromosomes. Once they separate these chromosomes, they pull them to the opposite ends of the cell, and then in telophase 1, now that they're separated in opposite ends of the cell starts to go back into its normal position, right? The nuclear envelope is starting to reappear, the spindle fibers are starting to disappear. Each cell that is being created here has half the number of chromosomes in the parent cell. So we're getting to the beginning here where we now have just one set of each chromosome in each cell. But this is only halfway. So now we do the exact same thing, but again, with less chromosomes. So each thing that I'm saying for the phase 2 of meiosis happens in 2 cells. So in prophase 2, right, we have 2 cells, but they're doing the exact same thing. So the centriole pairs duplicate again, they move to opposite ends of the cell, your nucleus never really fully formed, but it's going away again, and then your chromosomes are already formed, so it's just like prophase, but again. And then in metaphase 2, the exact same thing happens, right? Your chromosomes line up in the middle along the equator, your spindle fibers are in charge of moving them along. And then in anaphase 2, you pull these homologous chromosomes apart and you pull them into their sister chromatids. So now you have singular sister chromatids that are being separated and moved to the opposite poles. This is important because we are moving into a new terminology. So sister chromatids are now what's being moved, and you can tell that they're only half of a chromosome. And then in telophase 2, the nuclear membrane forms again, cytokinesis occurs, which is the separation of the cytoplasm, and now you have 4 daughter cells, which each have a haploid number, right? Half the number of chromosomes in humans, that's 23, and they are all genetically unique. You can see that in here that none of these cells are exactly the same. They're all just a smidge different, which is cool. That's like a really cool thing that all species are able to do, is create these genetically unique individuals. So it's interesting. This is just kind of going over it again. In interphase, you have your homologous pairs of chromosomes. They replicate. Now you have your homologous pair of replicated chromosomes, which then go through meiosis 1, where homologous chromosomes separate, and then meiosis 2, where sister chromatids separate. This is like a very abbreviated explanation of what I just went through, but it's a good overview. The last thing I want to talk about is crossing over. This is a, how do I want to say this? This is a way to produce genetic variation. This is a way that continues to make uniqueness in each of the cells. And in prophase 1 is when crossing over happens. It will not happen anywhere else, only in prophase 1, and it doesn't happen to every chromosome, but some chromosomes it will happen. So when the chromosomes pair up in prophase 1, and you have, your one from mom, your one from dad here, sometimes they will kind of like loop together, as you can kind of see in this part right here, and they will exchange genetic information. So what happens is, like you can kind of see in the second image, that the red and the blue are twisting up, and they'll just twist right off and connect to the other one. And so now you have a little bit of dad's information on mom's chromosome and a little bit of mom's information on dad's chromosome, and it gives you just a new twist on the chromosomes that you had, which just leads to more genetic variation, which is always what you want. The less genetic variation you have, the more inbred you are, and the easier it is to get genetic disorders or show genetic disorders, and the easier it is like to get sick, anything like that. So the more genetic variation, the better for everything we're going to talk about from here moving on. All right, great job. I will see you in class.